Protein sequence and structure in a medical context. So aims and objectives for this session are to look at how an understanding of sequence and structure helps us to understand and potentially treat disease. Can we understand the functional role of mutations in disease? How can we identify novel drug targets? And how can we engineer proteins for human therapy? Just to look very briefly at some of the applications of bioinformatics in disease. There are things like diagnosis, looking at mutations and looking at whether those are likely to cause disease, looking at prediction of disease risk, prognosis, things like in cancer, mutations in certain proteins and in certain regions of certain proteins are more likely to be linked with a bad outcome. Customized responses to disease, the idea of pharmacogenomics, identifying drug targets, and engineering proteins for therapy. And of course, in these days of COVID-19, there have been huge applications of bioinformatics in tracking the virus, tracking mutations and therefore strains of the virus, and understanding how it evolves, understanding what mutations may lead to evasion of the immune response that's been brought about by vaccines, and many, many other applications. Professor Orengo's group at UCL has been looking at comparing the ACE2 protein, the receptor for SARS-CoV-2 virus, across a number of species, including various house pets, to look at how many of those can possibly be infected by the SARS-CoV-2 virus because of similarity in the ACE2 sequence. This lecture is going to be divided into 3 parts, and in part 1 we're going to look at mutations. So what is a mutation? How do we define a mutation? Well, it's any change in the DNA sequence. But generally what we're referring to in this context is changes to DNA bases rather than larger changes and modifications such as rearrangements and so on. Now, an allele we define as the alternative form of a genetic locus, and of course we inherit one allele from each parent. For example, looking at the eye colour locus, we have the brown and the blue alleles. We also need to define a polymorphism, which is strictly a genetic variation present in at least 1% of a normal population. Now, of course, defining normal becomes quite difficult, and quite often the term single nucleotide polymorphism, or SNP, is misused simply to mean a mutation, and in particular to mean a mutation that doesn't lead to disease. Now, as we've discussed before, around about 2% of the genome codes for proteins. So if we define a gene as the coding region plus 10 kilobases upstream, because we often get control regions upstream of the coding region, then at least 98% of genes are within 5 kilobases of a SNP. 93% of annotated genes contain a SNP. 59% of genes have at least 5 SNPs, and 39% of genes have at least 10 SNPs. So you can see that these polymorphisms really do occur very regularly within coding regions of proteins and within the upstream regulatory regions. Now, SNPs, polymorphisms, are really quite useful in a number of ways. We can use them in understanding evolution, so some alleles may be advantageous in one environment but disadvantageous in another, and therefore we can track the presence of certain alleles throughout evolution and throughout the spread of populations throughout the world. They're also useful in DNA fingerprinting, And of course, in areas like forensics, where we can use DNA fingerprints to identify people being present at a crime scene, they can be used as markers to map particular traits, such as diseases and characteristics, and they can be applied in pharmacogenomics for genotype-specific medications. So what effects do SNPs have? Well, NS SNPs, in other words non-synonymous SNPs, result in what we could refer to as a single amino acid polymorphism, or SARP. Now, using this looser definition of a SNP and, by analogy, a SARP to mean simply a mutation, then such things may affect function, either directly or affect targeting, in other words, where a gene product ends up going in the cell, and may affect things like post-translational modifications. Any type of SNP, whether non-synonymous or synonymous, may also affect splicing and affect things like transcription. So let's look at some of these applications. First of all, SNPs as markers. Again, we'll define a few terms. Linkage is how close two loci are on a chromosome. And linkage disequilibrium, which refers to alleles rather than loci, refers to the fact that alleles, or SNPs, which are co-inherited more often than expected, are closer together on the genome. That means that we can look at the co-inheritance of a particular SNP with co-inheritance of a particular condition. Now, that doesn't necessarily mean that the SNP is causative of that condition, but that it is close to a locus on the DNA which is causative of the condition. A haplotype is the set of alleles on a particular chromosome. Now, a given population may have a high incidence of a particular disease, and as I said, if we can find SNPs that are in linkage disequilibrium and correlate with the disease phenotype, we can use these as markers. So let's assume there are two SNPs and one gene that are associated with a disease. So we've got two populations over here, population 1 and population 2. And in population 1, we have a large fraction having this genomic DNA, in other words, allele big A, and a smaller fraction being heterozygous, and a very, very small fraction having the disease-causing alleles as homozygous. In another population, however, that can change a lot. So here we have a much larger fraction of people who are heterozygous, and also a larger fraction of people who are homozygous for the disease condition. Now, if we have these two SNPs that are either side of these two different alleles, we can identify those SNPs, identify the fact that they're in linkage disequilibrium, and link those to the disease status. Polygenic traits, such as cancer and Alzheimer's, can be treated in a similar way, and here we tend to talk about QTLs, quantitative trait loci, and a quantitative trait is a continuously variable thing. In order to analyse these, though, we need very large populations. So, in the example that we just looked at for a monogenic disease, where we have three combinations of AA, A little a, and little a, with one gene being involved in the disease, we have these three. So if we have n genes, we'll have 3 to the power of n different combinations possible. And of course, this sort of thing can be applied not only to diseases, but also to things like behavioural traits. Let's look now at disease-causing mutations. So, as I said, these are not strictly SNPs or polymorphisms, but quite often the term can be misused in that way. A better term to use is really SNVs, or single nucleotide variations. Now, some of these SNVs are potentially lethal, and this idea at least was first described some 2,000 years ago by Lucretius Caro, who said, "What is food to some men may be fierce poison to others." Now, of course, we don't know what he was referring to, but it's possible he was referring to this condition known as Favism. So this condition leads to the lysis of red blood cells when eating things like fava beans, or broad beans as we call them here, and other foodstuffs. This affects around about 10% of the world's population and results from a faulty glucose 6-phosphate dehydrogenase. Now, this is a cytoplasmic enzyme encoded on the X chromosome, which of course means that males are more likely to be affected by this condition than females. Now, G6PD produces NADPH, and that's used to regenerate glutathione, which maintains the reductive state of the cell. Now, in most cells, there are other routes to producing NADPH, but in red blood cells, this is the only route, because remember that red blood cells don't have a nucleus. Consequently, when exposed to oxidizing agents, including some of the ingredients in these beans, and also a number of drugs, patients end up with lysis of red blood cells and what's known as hemolytic anemia. This is actually a very important disease because some of the drugs that can cause this problem include the antimalarials, and this Favism condition does tend to be quite common in populations where malaria is a problem. Now, interestingly, there is also some evidence that these mutations in G6PD also decrease the likelihood of malaria infection. Now let's just look at a few examples of single nucleotide variations in G6PD. One of them here, 376A to G, leads to an asparagine to aspartate mutation, but this leads to essentially normal activity, so could really be considered to be a single nucleotide polymorphism. And this is present in about 20% of African males. This mutation, leading to valine 68 methionine, leads to 90% activity and is present in about 20% of African alleles. This mutation, serine 188 phenylalanine, leads to zero activity, and that's present in 20% of Mediterranean Caucasians. There are also many other mutations known. This is just a small sample to give you an overview of the differences in effects that these mutations can have. So what about genetic testing? If we've got this information about mutations, is that actually a useful thing to have? So it depends on the particular condition. Certain mutations are of very high utility. So if we know about these mutations in G6PD, then we can largely control the diet and be careful with medications in order to avoid these rather unpleasant side effects. Similarly, with this condition, multiple endocrine neoplasia type 2, if we know that a patient has the mutations that are related to that, then we can perform a thyroidectomy, and that will basically stop the cancer from developing. At the opposite end, certain things like Alzheimer's disease, knowing that you have a mutation that is going to increase your chance of having Alzheimer's disease, may actually be a problem because there is no effective treatment. And therefore, people can be in a condition where they're expecting the worst to happen. And then in the middle we have things like mutations causing hemochromatosis, where phlebotomy can be useful, mutations causing colorectal cancer, where we can make sure that people are screened very regularly in order to be able to perform surgery early on, and things like mutations leading to breast and ovarian cancer, where again prophylactic surgery can be useful. Okay, that brings us to the end of part 1, and in part 2 we're going to look at customised response to disease and the identification of drug targets.